Good morning, sir.
Good morning, sir.
Good morning. So exciting time ahead. A big thanks to everyone joining our awesome Heredity Biosciences webinar. Today, goal is to spread awareness, and we promise more life sciences events like this in this future. So firstly, I super warm welcome to each and everyone's today's special webinar. I am Dr. Gopalakrishnan Purohit, CEO and co-founder of Heredity Biosciences. I'm really happy to have you all here with us for this fantastic webinar today. So first of all, thank you, Susri, for this wonderful introductions. Today's webinar expertise topic is characterization of tumor cellular heterogeneity using single-cell RNA sequencing. So this is discovery the revolutionary insight into frontline technology provides and revealing the complexity of cellular diversity within tumors. Join us for an informative investigations for the forefront of biosciences innovations. Now, allow me to extend my deepest gratitude to our esteemed guest speaker, Dr. Sillarine Kurkalang, research professional at University of Chicago Medicine's Comprehensive Center, United States. We are immensely grateful for the acceptance of our invitations. Thank you. The entire Heredity Biosciences team is genuinely appreciative for your presence and expertise. Thank you, Cellular ma'am, for your valuable contributions to today's webinar. Now, audience, today's webinar is centered around the interesting study of tumor cellular heterogeneity using single-cell RNA sequencing. So join is on unraveling the complexities within a tumor at the single cell level as we explore the groundbreaking insight provided by this fraud sequencing technology. Fasten your seatbelt. So for an enlightening journey through the landscape of cellular diversity within the tumor, cracking light and a crucial aspect of cancer research. So to know more about this, what is tumor cellular heterogeneity, and today's topic highlights is the tumor heterogeneity, a defined characteristic of cancer, poses challenges to effective therapy and fuels tumor progressions. Examining both inter and tumoral heterogeneity not only yield insight into the tumor development but also inform the creation of personalized treatments. While previous high throughput sequencing technology offers is imposed into the tumor ecosystems, they lack the resolutions to capture a detailed cellular landscape. But recent advancement in the single cell technologies now provides an unprecedented resolutions profiling transcriptomics, genomics, proteomics, and epigenomics along with the spatial distributions. This breakthrough accelerate basic and translational research, cancer research, uncovering the intratumoral heterogeneity. Crucial, it has observed to the cancer cells and transcription between the states, adapting to the dynamic and tumor microenvironment, enhancing to the cellular plasticity, amplifying tumor heterogeneity, and we will understand the molecular mechanism governing to this cancer cell plasticity is vital role for the development of the precision therapies. Now, once again, thank you, Dr. Sillarine Kurkalang, for gracing with us your presence today. We greatly appreciated your times and knowledge. Now, audience, before we begin to our webinar, as we introduce our before you introduce to our esteemed guest, allow me to provide a overview of Heredity Biosciences. Heredity, name established in 2018 and rebranded as a Heredity Biosciences in 2021. We have evolved in a prominent provider of research and training services for hospital and life sciences institute and pharmaceutical firms. It is a trusted name of medical and scientific community. Our esteemed reputations stem from the session team of scientists, research professionals, backed by the robust national advisory board. We have and a diverse team. We specialize in genomics, proteomics, and metabolomics and bioinformatics. We are committed to the quality. We service frontline technology to deliver the cost-effective solutions Not only for this India, we are solutions we will spread in the globally. Our recent collaboration with the Heric Life Sciences Private Limited promises exciting advancement, aiming to diverse impactful innovations in life sciences and healthcare professionals research. Now, Heredity Biosciences extend to locations. Primary location is, as you know, that Mayapur Lagoon Road in Bhubaneswar, and our new branch last year opened into the Guwahati, Assam, inaugurated. It is in last year. This expansion is a significant milestone to symbolizing the progress of our growth, not only for the strength of capability, but also open our door for valuable collaboration with the local institutions, professionals, scientists, and for the life sciences professionals. We are genuinely excited to the potential growth being preceding for the meaningful contribution to advancing to the life sciences field. So, Heredity Biosciences provided for the specialized services of life sciences sector and healthcare sectors. So, focus on the research and training services. Our life sciences service, as you know, that as a genomics, proteomics, and metabolomics, with advanced facility ensuring the national-wide accessibility. In healthcare research services, we explore to the vital domains such as clinical microbiology, infections genetics, cancer genetics, fertility genetics, and conventional PCR-based DNA studies. For detailed information, everything is inside our websites also. So now we move on highlight to the training part of Heredity Bioacademy. So as Odisha's leading training institute, we are proud to offer comprehensive hands-on experimental research training to the students. Students as well as the professionals also. These are the listed course modules provided complete hands-on procedures. So some specialized module introduced to this to our mm, to our listed below. So this is a clinical embryology, that is a male infertility part we are focusing for that, and histopathology and histochemistry. qPCR based how we will diagnose to the cancer genetics, so that we have to focus to it, and we are the frontline modules for design to the address to the growing need for the advancement in the field of the biosciences. Heredity Biosciences is excited to announce the comprehensive dissertations programs also and personalized for the life sciences students. This program offer unparalleled hands-on exposure to advanced experimental laboratory practice, providing the holistic learning experience in the field. So students enrolled in these programs will research with the advanced research into the frontline experimental technologies, gained individual practical knowledge, one-to-one knowledge that complete their academic learning. This important is on development a deep understanding of advanced laboratory practice within the life sciences domain. Heredity Biosciences is committed to provide a dynamic and enriching environment that rising to the next generation of life sciences. Sorry. So this dissertation program stand as a gateway to acquiring essential skills and expertise, preparing students for the challenges of opportunity to the job sector and the research sectors to evolving the landscape of biosciences. So join us the Heredity Biosciences and embark to the journey that combine to the academic accuracy with hands-on applications, empowering to your become a future leaders into the field of the life sciences. So we have a good infrastructure, so facilitating both training and research part, and we have invited to everyone to visit our central facility and witness to the following structure: world-class centralized air conditioning facility, high conductivity Wi-Fi environment, complete research learning facility, well-trained, experienced training trainer and demonstrator, complete hands-on individual exposure, one-to-one exposure. I am personally guided to the students, publication assistance, placement assistance also for the students. Over last 3 to 6 months, we are really achieved for the placement activities facilitating to the students, resulting in the numerous success stories. So many students have completed the interview with the esteemed organizations. They are going to placement opportunity, are still undergoing to the for other students. Last 3 weeks, these are the last 3 weeks. Recent placement status showing to the slides. Some of the students already have a placed, but we are not prepared to the things. But we are congratulate to those complete students and wish them to the prosperous and fulfilling the career journeys ahead. Heredity Biosciences, as you know, that we are focusing to the research part, so that we are published every month should be publications. So last 1 month we are published with 2 to 3 publications, but in the overview overall, more than of these 25 to 30 publications and 3 patents we have already. So these are the milestones. So Heredity Biosciences recently come together. So second Foundations Day to interns with our interns to joy celebrate to our second Foundations Day. These are the things. These are the pictures which represent to the all these activities. The promises made to the inception of Heredity Biosciences were beautifully throughout the day festivals held to our office. It is a wonderful event, filled together, friendship, and sad to the moment of the joy. Um, December 18, we are celebrated to that. The entire day was a celebrations, and everyone present thoroughly enjoyed the occasions. Thank you all to who contributed to the making to this event so memorable and fruitful. So now, ladies and gentlemen, now I am thrilled to introduce our distinguished guest speaker for today's webinar. Uh, Dr. Sillarine Kurkalang, currently serving as a research professional at the University of Chicago Medicine Comprehensive Center in Chicago, United States. Dr. Sillarine's academic journey is a testament for relentless pursuit of excellence and unwavering dedication in the field of cancer molecular genetics. So Dr. Sillarine and her PhD in cancer molecular genetics from Northeast Hill University, Ceylon in 2015. Her pioneering research investigating into the loss of heterogeneity at chromosomes 4q27 and 3p15 regions, investigating the methylations and expression status of selected tumor suppressor and checkpoint genes in betel knot induced to human and mouse samples. Prior to her doctoral studies, she completed her master degree in biotechnology at Bangalore University in 2007. Dr. Sirali's professional journey is characterized by key roles in significant research endeavors that spanned various critical projects. She committed her path as a research associate at the National Institute of Biochemical Genomics, where she completed 2016-2018. She focused on identifying recent somatic mutations in gastric adenomas presenting within the major populations in Northeast India. Continuing her impactful research, she assumed the role of DBT Research Associated 1 and 2 from January 2019 to December 2020. We concentrated on unerring intra-tumor heterogeneity associated with a tumor recurrent in oral squamous cell carcinomas, giving buccal samples. Subsequently, as a principal project associated with the February 2021 to October 2021, Dr. Sillarine significantly contributed to multiple dimensional research to enabling system medicines, accelerating using a cluster approaches. Her journey is translated to serve as a research professor at the University of Chicago Medicine's Comprehensive Cancer Centers from August 2023. Her expertise span a diverse skill set encompassing target sequencing, single-cell RNA sequencing, and interesting professionals in animal cell culture in involving primary cells and cell lines. Dr. Sillarine dedicated to advancing scientific knowledge in underscoring by her contributions, which extend more than eight outstanding publications in prestigious national and international journals. Today, Dr. Sillarine will say to the invaluable sort into the webinar titled Characterizations of Tumor Cellular Heterogeneity using Single-cell RNA Sequencing. We eagerly anticipate the wealth of knowledge she will impact. Thank you, Dr. Sillarine, for joining us and enthusiastically looking forward to the enlightening and enriching session ahead. So this is the team Heredity Biosciences team. So they are truly appreciated. My team members, the best effort of the research and students, training activities. Thank you. Now we'll start for this session for this Dr. Sillarine. You will start for the presentation.
Okay. A very good morning to everyone. First, I would like to thank the Heredity Biosciences for conducting such a webinar that I believe it will be helpful for many. And I'm thanking for your kind words and your long introduction. I'm very thankful. And thank you for all the participants for giving their time in attending such webinar. Just sharing the slides. Is my slides visible?
Yes.
Yes. Yes, ma'am.
Yes,
Okay.
ma'am.
Yes, ma'am. Okay. Today I'm going to talk on the characterization of tumor cellular heterogeneity using single-cell RNA sequencing. As you know that every tissue in the body is composed of multiple cell types. For example, the adipose tissue consists mainly of the adipocytes, but there are also other cells as well, like the macrophages, the endothelial cells, the lymphocytes, the stem cells. So all these cells together, they constitute the adipose tissue. Another example of a complex tissue, which is the blood, consists of, again, multiple cell types, the red blood cells, the platelets, the white blood cells, and each cells, they are different from each other. They are different in their morphology, different in their functions. They do exhibit a distinct gene expression, distinct genomics and epigenomics profile. However, only until recently, most of the molecular studies that have been studied have relied on the bulk analysis. So the precise characterization of each cells or the cell types and the cell state is usually masked by bulk genomic methods. For example, if you want to study the gene expression of a particular tissue, as you know that each tissue consists of multiple cell types. So with bulk method, the gene expression profile that you will get at the end of your experiment is actually the average gene expression profile from all the cells, not actually the actual gene expression, but the average of the gene expression that obtains from all the cells. Thus, it masks the cellular heterogeneity, where you will not be able to get the actual representative gene expression from individual cells or the tissue as a whole. However, this challenges has now overcome by the advancement of the technologies like the single-cell sequencing technologies, where with single-cell sequencing, you'll be able to get the gene expression, we'll be able to get the gene expression. When we talk about the gene expression, we will be able to get the gene expression profile of individual cells, as each cells have their own distinct gene expression profile. They do not have the same expression profile. They do not have the same expression level of a particular genes. They do exhibit their distinct or the different levels of expression for each genes across the cell types. Thus, when we get the expression profile of all the cell types, it helps in revealing the heterogeneity and also within across thousands of cells or within the population of cells, we will be able to get it will reveal the heterogeneity of the cells or across the population of cells. So in this way, you'll be able to get the actual representative gene expression profile of a particular cell or particular cell types. That is actually the what is actually happening inside the tissue. So how does single cell sequencing work? It works by partitioning each individual cells of a particular samples that you have provided, and then it will generate, when we talk about the gene expression, it will generate the gene expression profile of each individual cells that you have used. So there are many technologies that have developed for this single cell sequencing. However, this is the most common use platform. This is the microfluidic chips here, where we will be using for generation of or single cell barcoding. So in these microfluidic chips, as you can see, there are rows. There are multiple rows, and in each rows, you will be adding the oil. You'll be adding the sample in the sense that a single cell suspension for every sample, the gel beads, and there is a row here where we do not add anything, but at the end of the generation of the at the end of a single cell barcoding, you'll be collecting the gel bead in emulsions or the gems. So once you load all of these requirements in the chip, these chips will then be loaded into the machine, and then the single cell barcoding will happen, and or the gel bead in emulsion called as gem. So then the gem generation will be happening in this machine. But actually, what happening inside is quite complex. Whereas I told you that the chip is a microfluidic chip, this is a multi-channel microfluidic chip. This is the inside the chip where you already added the gel bead. So the gel bead will flow from one of the channel, and along the channel, these cells and the enzyme will come from the other channel that are perpendicular to the main channel. It will meet the bead, and along the way, the oil that comes from another channel, once the oils meets the bead and the cells, the aqueous solution will partition the oils into an oil droplets. So each oil droplets contain one cell, one bead, along with the RT reagents and the enzymes. So in this way, this process is it keeps continuing until the completion of generation of these oil droplets, also called as gel bead in emulsions or the gems. So after the gem generation is completed, we will take out this chip from the machine, and then we will again from this row where we need to collect the gems. So we will collect the gems in the tube. So now in each tube, you have multiple thousands of gems, where each gem, as I told, contain this gel bead, the cell, and the RT reagents and the enzyme, which is required for generation of the cDNA for each particular cell. So once we collect the gems, we'll then incubate in thermocycler for reverse transcription, where it will generate the cDNA inside the gem. So each gem have now the cDNA from a particular cell. The next is that we need to, since now the cDNA has come from each cells, and already the gel beads that we have added contain the barcode. This is other sequence that helps us in identifying. Using the barcodes, we will know that this cDNA comes from which cell. So it helps as an identifier of a particular cell. So once the cDNA was generated, we can now, since we already has the barcode, so we need not to bother about cDNA comes from which cells. So we can now pool all these cDNAs. So for pooling cDNAs, we need to break these gems. So for breaking these gems, we will add the recovery reagents. Recovery reagent, it will break the gem and release the cDNA into the solution. So when it releases the cDNA, we will then amplify the cDNA and use that amplified cDNA for generation of single-cell sequencing libraries. So when the libraries was generated, then we will then use this library for sequencing. And once the sequencing was completed, it will generate the sequencing data containing the gene expression profiles of thousands of cells uh, for each samples. Tumors are very complex. They are the complex ecosystems. As you can see here, this is the tumors, but inside these tumors, they are very complex. They contain not only the tumor cells, but it contains the fibroblasts, the T cells, the macrophages, the dendritic cells, the tumor cells as well. And as I mentioned earlier, that each cells are distinct from each other with respect to their morphology, the functions. They have their distinct genomic and epigenomic profiles, which this heterogeneity within the tumor will influence the tumor growth, the response to therapy, the recurrence means it depends on what particular cell type with particular profile they have. It will influence that the tumor can recur or not recur, and it will respond or not respond to a particular therapy. Some particular cell population, they do respond to some therapy, but some other population of cells, even within the same tumor, they do not respond to therapy. So even when we see the patient, some patients, they initially they do respond to some treatment, but eventually after sometimes the cancer relapse or they become resistant to treatment. This is because due to this kind of heterogeneity that are present within the tumor, where some population of cells, they do respond to treatment, but the presence of some population of cells that are resistant to treatment, and when those resistant cells were continue to proliferate and increase, then later on they do not respond, the patient do not respond to a particular treatment. And some cells helps in promoting the tumor growth and metastasize. Also, due to this such heterogeneity within the tumor, it also influenced the survival periods across the patient. So here today we're talking about the oral cancer of the gingival region. Why the oral cancer of gingival region, which is also known as oral squamous cell carcinoma of the gingival region. This is because this oral cancer of the gingival region is the most common cancer among men in India, having poor prognosis and also high tumor recurrence. So in this particular study, our main objective was to profile the nature and the extent of cell type and the cell state diversity in OSCGB. So for this, we generate the single-cell RNA sequencing profiles for the primary tumors from oral cancer patient, where we collected the tumors from the treatment-naive patient, means the patients that who have not got any treatment. So we collected the tumors. The tumors was collected from these patients, the oral cancer patient attending at the Dr. R. Ahmed Dental College and Hospital, Kolkata. After collecting these the tumors, then we immediately transport into storage solution and on ice, and immediately transport to the lab. So once we reach the lab, this tissue, we need to dissociate the tissue so as to obtain the single cells. So after tissue dissociation, and we got the single cells and these cells, then we need to assess for the viability. Assessing the viability means how many of cells are the how many cells are live cells and how many are the dead cells. So only those samples that contain more than 70% of live cells can be processed further. And also we perform the cell counting, so we know like how many cells are there per samples. So we select a particular number of cells to use. It depends on each and every experiment, but for us, we use 5,000 cells. So we use these 5,000 cells from each samples, and then use this for single cell barcoding or the gem generation and generation of single cell RNA sequencing libraries. So once these single cell libraries were constructed, these libraries were then we assess the quality of these libraries, and then we proceed for a sequencing where we sequence in the NovaSeq 6000. After sequencing, the sequencing data thus generated, we will be analyzed those data using the cell ranger, where the BCL files will be converted to the FASTQ files and then to the count matrix, and those count matrix will then be further analyzed using the R toolkit, which is the CPAT. Upon analyzing the data, we after clustering these cells, now we need to identify that which of these cell population or the cell clusters belongs to which cell type. So using the known marker genes that are well established, so we use those genes to identify which cell types each clusters belong. We use many of the markers. I'm showing here just few of the markers. Like for example, as you can see figure in the right, KRT14 was one of the marker we used to identify the epithelial cells, since it's this the epithelial marker genes. So as you can see that the expression of KRT14 is highly expressed only these particular clusters. Thus indicates that these clusters can be classified as the epithelial cells. So similarly, this particular cluster shows high expression of the T cell marker, which is a CD3. So this particular clusters we classified as the T cells. So similarly, we classified the B cell clusters, the myeloid cell clusters, mast cell, fibroblasts, and the endothelial cells. And as you can see that with this circle plot here, you can see with different colors. Each the colors indicates the cell from the patient. Like the red colors cells come from patient 1, the orange from patient 2. You can see that the size difference because these are the fractions of cells. So even though a particular cell types comes from all patients, but they do have variability or differences in the percentage of cells for each patient. Next, we then classify the malignant and non-malignant cells. As you know that this is the epithelial tumors, so the non-epithelial cells, we know that these are the non-malignant cells. So we extract only the non-epithelial cells and then reclustering them, and we observe that these particular clusters again belongs to particular cell types. Like these clusters belongs to the T cell, the fibroblast, the endothelial cells. And as you can see the figure on the right, each dot is a cell and the color by patient. As you can see, the light blue color is the patient cells comes from patient 10, the red color cells come from patient 1, and so on. And then you can see that for each particular cell types, all cells comes from all patients. Thus, it shows that the non-malignant cell clusters were cell type specific. We next, so now the epithelial cells are there, and then we know, even though that this is the epithelial tumors, but not all the epithelial cells, they are the tumor cells. So we want to use only the exactly tumor cell, which is the malignant cells. We then classify the malignant and non-malignant epithelial cells, and by using the in-house developed method. We then classified and we able to identify around 7,000 of the epithelial cells were the malignant cells, and then clustering those malignant cells. We found that unlike the non-malignant cells, These epithelial malignant cells, they cluster mostly patient-specific, not cell-type specific. As you can see here, this is the clusters or cell clusters of the epithelial malignant cells, where each clusters like clusters in green, in light green, blue, light blue. So these clusters belong to cluster 2, cluster 5, cluster 4, cluster 11. So these particular clusters, these particular cells of these particular clusters, they belong to one patient. As you can see the figure in the right, it belongs to a patient number 8. Similarly, cluster 3, which is dark green in color, this clusters comes from cells from patient 12, as you can see in figure right. Cells coded in the yellow color that comes from patient 12. Similarly, cells in cluster 7, cells in cluster 1, all these cells, they cluster together. These cells comes from individual patients. However, there are few clusters, like here, these clusters, like the cluster 6, cluster 8, cluster 14, 15, 16, they do contain cells from multiple patients. So those clusters are important as they represent the cells from the patient. As you can see here in this figure, you can see that we have grouped them in group 1 and group 2. Since when we look carefully, it looks like these particular cells from the group 1 and cells from the group 2, they cluster a little bit separately from each other. Then and this is not very usual. And then when we go back and see the clinical information, we find that these particular cells from group 1, they belong to patient who has oral submucous fibrosis, whereas the cells from these clusters in group 2, they belongs to the patient who do not suffer from oral submucous fibrosis, thus indicating that cells with OSMF have distinct gene expression profile compared to the cells without OSMF. We then look a little bit, into details of the malignant cells, and when we did the differentially expressed genes, and then we identified the significantly expressed genes. Using those significantly expressed genes across these clusters, we then try to find which of the pathways were enriched, and we find that 40.61% of all the malignant cells were enriched with the expression of the gene signatures that are known to be involved in EMT, like the ECM receptor interaction, the focal adhesion. This ECM receptor interaction focal adhesion was reported to be involved in development of EMT in multiple cancers. Like as you can see the figure on the right, cluster 1, cluster 3, cluster 6, cluster 14 and 15, they were highly enriched with cells expressing these ECM receptor interaction and focal adhesion. Thus indicating that these cells might have undergone epithelial to mesenchymal transition. So next we then see if they really express the epithelial to mesenchymal transition signatures. We found that really those particular clusters were really enriched with cells expressing these epithelial to mesenchymal transition. However, when we individually check for the expression of the epithelial and mesenchymal marker genes, we found that they still express both the epithelial and mesenchymals, indicating that they have not fully transitioned to mesenchymal, but they might be still in the intermediate phase or the transition phase, which is also known as the partial EMT. So we check for the partial EMT gene signatures, and we find that these particular clusters, they were enriched with the partial EMT gene signatures. which confirms that these are, which again indicates that these are the partial these cells undergoing partial EMT. But what you see, there are other clusters that are not enriched with these EMT. So we're wondering what signatures were then enriched by the cells in these other clusters. We look for the expression of the their cell type signatures, and we observe that 8.6% of the malignant cells were enriched with the fetal signatures, including the fetal squamous epithelial, fetal lung squamous epithelial cells, the fetal stomach squamous epithelial cells, cluster 7, cluster 8, cluster 10, cluster 12, and cluster 16. These cells were not enriched with EMT signatures, but they are enriched with fetal signatures, indicating that these are fetal-like epithelial cells. Now, the fetal signatures were known to be expressed only in the fetal cells, but however, in the tumor cells, there are lots of cellular reprogramming where the cells do express genes that were involved in the early developmental phase, like the fetal development. So these signatures were also reactivated in the cancer cells to further enhance the tumor growth and progression. We also observe, apart from the EMT and the fetal cell type signatures, we also observe that these particular clusters of cells, they do express particular genes like the cancer testis genes. So again, the cancer testis genes was only expressed in the germ cells, but then they do reactivate in the tumor cells and re-express in the tumor cells. And we found that these cancer testis genes were expressed in the subpopulation, not all the cells, but only in the subpopulation of cells. So taken together, so these particular cluster indicatives of the germ-like epithelial cells. Taken together, we found that cluster 0, cluster 1, cluster 3, cluster 6, 14, and 15, these clusters consist of cells that were enriched with expression of PMT signatures, but showed lower expression of other signature or do not express other signatures. Whereas cells in cluster 7, 8, 9, 10, 12, and 16, they showed a higher expression of fetal signatures, but lower expression of PMT. And the other clusters which do not express PMT or fetal signatures, they showed higher expression of cancer testis genes, including cluster 2, 4, 5, and 11. Thus indicates the mutual exclusiveness in the expression of these genes in the oral cancer. We then look across the patient and we find that there are some patient like patient 1, 2, patient 9, patient 10, patient 12. They enrich with cells that shows high expression of PMT signatures. Whereas other patient like patient 3, patient 4, patient 6, patient 4, 15, 14, 16 were enriched with fetal cell type signatures. However, only one patient which was not enriched with fetal cell type signatures or PMT signatures, they were enriched with cancer testis genes. Thus indicates the importance of partial EMT and fetal cell enriched expression of fetal signatures indicate fetal cellular reprogramming could plays an important role in the oral cancer. So EMT and fetal cellular reprogramming could play a very important role. However, we did not conclude much on the cancer testis genes as we have only one patient. Maybe we need some more patient to study to come into conclusion. Although it indicates that it also indicates that it could place and also an important role in the oral cancer of the gingival regions. Apart from the malignant cells, we also look into the other cells in the tumor microenvironment. We found that most of the cells are the immune cells, like higher proportion of immune cells compared to the strong cells in the tumor microenvironment. And among these, 36.4% are the T cells and 14.7% are the myeloid cells. Whereas the stromal cells like the fibroblasts just 5.1% and 3.1% of the endothelial cells. Thus indicates that the importance of the T cell and the myeloid cells in OLCCGB. When we look into the expression heterogeneity of the particular cell type, like the T cell, we find that there is variability in the proportion of these T cells, where the T cell, like patient 12, patient 13, they have just less than 10% of the T cell, whereas patient number 10, patient 5, they have more than 50% of the T cells. So there are lots of heterogeneity even in the number of the T cell within the tumor. When we look a little deeper to further classify the T cells, we again use the known T cell markers. The major one are the CD8 markers and CD4 markers. So we want to see which are the CD8 T cell and CD4 T cells. So these CD8 are highly expressed in these clusters, which is this cluster 1 and 7, and also this cluster 3 and some cells in the cluster 5. Whereas CD4 positive T cells are mostly the cluster 4, cluster 8 here. And then when we look again little further details using the cell type, cell state signatures like the cytotoxic T cell and hosted T cell marker genes, we identify that this particular cluster 1 and cluster 7, as you can see in the heat map on the right, cluster 1 shows higher expression of the cytotoxic markers, and also similarly with cluster 7. So these are the cytotoxic CD8 positive T cells. Whereas you can see this is cluster 0. Cluster 0, you can see the heat map. You can see that these were enriched with the naive T cell markers indicated these are the naive T cells. Whereas this is cluster 4. You can see the cluster 4 shows higher expression of the Treg markers like the FOXP3 and the IL-2RA, indicated that this population is the Tregs. This particular clusters, this light blue colored in the light blue, this particular cluster, cluster 5, as you can see in the heat map, it shows higher expression of the proliferation markers, the top 2A and the FTI67, indicated that these particular clusters, they are the proliferative T cells. Similarly, the cluster 8, cluster 3, as you can see here, they do express. There are cells that shows higher expression of the exhaustion marker, thus indicates that these cells are the exhausted T cells. And there is particular clusters, as you can see, these light green clusters and the small cluster which is pink in color. So these particular clusters, they are neither CD8 or CD4 T cells. So they are CD8 and CD4, CD8 negative, CD4 negative, also known as double negative T cells. So these T cells, we observe this higher proportion of these T cells also. We observe around 36 to 65% of the double negative T cells. And these double negative T cells are reported to plays an important role in the tumors where they have their strong anti-tumor effect. These T cells, they involve in enhancing the anti-tumor immune response. Looking across the patient-wise, the enrichment of the cell states, T cell states, we find that patient 9, patient 5 and 15, they were enriched with the cells were enriched with cytotoxic markers, indicative that they were enriched with the cytotoxic T cells, whereas patient 8, patient 12, and patient 2, they were enriched with the exhausted T cells. However, there are 3 patients, patient 10, patient 1, and patient 3, they were enriched with both the cytotoxic as well as the exhausted T cells. So looking in just one population, also there is lots of heterogeneity within the population of T cells, heterogeneity in the number of T cell across patient, and the heterogeneity of the T cell states across the patient, and even within the same tumor. The next population we are looking is the macrophages, and when we look to the macrophages, this is the clusters of the macrophages, and these cells, macrophages, shows higher expression of the pro-inflammatory and the anti-inflammatory markers, suggesting the presence of both the M1 and M2 macrophages. And when we see the expression of the M1 and M2 macrophages, we observe that these macrophages really both the M1 and the M2 markers, thus indicates that the macrophages here are there is an intermediate M1 to M2 macrophage polarization in OSCGB. Previous studies from NIBMG itself, previous studies, they have identified the significantly mutated genes that are specific to OSCGB. As you know that oral cancer site in India is different from oral cancer site in the West. Oral cancer site in the West is mainly the tongue, whereas oral cancer site in India is only the gingival buccal. So they've identified the altered genes that are specific for the oral cancer of the gingival buccal region, including ARID2, which is AT-rich interaction domain 2, MLL4, USP9X, UNC113C, TRPM3. ARID2, which is a chromatin remodeling genes, it belongs to the PBAF complex. So here is the ARID2. It belongs to the PBAF complex, which is a subunit of the SNF complex, known to be involved in chromatin remodeling and transcription. So these genes, this is the main components of the PBAF complex, along with the BRD7 and PBRM1. Many studies have reported that the deficiency or the lack of function of these chromatin remodeling complex enhances anti-tumor immune response. So in one study that showed that inactivation of these genes like the ARID2, PRD7 and PBRM1, the melanoma cells that with inactivation of these genes, they are more sensitive to T-cell mediated cytotoxicity, and also enhance the secretion. There is an enhanced secretion of the chemokines that recruit these T cells to kill the tumor cells. The next study shows that this ARID2 expressions is negatively correlated with T-cell cytotoxicity markers in many cancer types. In the sense that when the ARID2 expression is high, the T-cell cytotoxicity marker is low. But when the ARID2 expression is low, the cytotoxicity markers expression is high. Another study shows that loss of function mutation of PBRM1. Patient with this loss of function, so the inactivation the inactivation of this PBRM1 through loss of function mutation, patient with this loss of function mutation, they showed improved response to when we treat, they showed more response to treatment when we treat with PD-1 and PTL1 in metastatic renal cancer patient, compared to those patients who do not have this mutation or where the PBRM1 is not inactivated. Thus indicates that it shows that the importance of these PBAF complex genes We then therefore characterize the expression state of this feedback complex gene in our single cell oral cancer. And we saw that there is cells that express and cells that do not express these genes. As you can see here, these cells colored in red are the cells expressing ARID2 or PBRM1 and BRD7, whereas the cells colored in green are the cells that do not express ARID2, PBRM1 or BRD7. And when we see patient-wise, I'm not shown here, but patient-wise also there is the variability again in the proportion. Some patients, they do have a higher proportion of cells expressing, whereas some patients, they have lower proportion of cells that are expressing these PBAF genes. Since previous these studies have reported that cells that do not express these genes are more response, they enhance the response, anti-tumor immune response, whereas cells expressing are resistant to immunotherapy, or they so we check for the immune resistance signatures, to those gene signatures that express by those cells that are resistant to immunotherapy. So we again that in our data also it shows that those cells that express these PBAF genes, they shows higher expression of genes that are resistant to immunotherapy. And we also check for the epithelial to mesenchymal transition signatures, and we also observe here that those cells that are expressing shows high expression of epithelial to mesenchymal transition markers compared to the non-expressing cells, indicating that these populations of cells, they might be resistant to therapy as well as involved in the growth of the tumor and metastasize in metastasis. When we did the differentially express, we find the differentially expressed genes between the cells expressing and the cells that do not express these PBAF complex genes. We observe that the cells that express these PBAF complex genes, they also showed they have significantly overexpression of genes involved in metastasis, promoting tumor growth, and also involved in the stem, like in inducing the stemness in the tumors, like the MERIT1 and the NEAT1. So again, it shows that these cells expressing the PBAF complex genes previously have shown that they are showed high expression of immune resistance, and means they might be resistant to immunotherapy, and they might also involve in metastasis and in tumor growth, and also they can involve in tumor recurrence. As you know that again, the cell cycle also is very important in the tumor growth, and where the deregulation of cell cycles helps in tumor initiation and progression of the tumor. We then evaluate for the, we check like how many of the which cell states, which cell cycle state do these cells in OSCC GB tumors, where we observe that majority of the cells and high proportion of these cells were in the G1 phase of the cell cycle. As you can see here, these cells colored in red, they are the cells in the G1. The cells colored in green are the cells in the S phase. Cells colored in the orange, they are the G2M phase. And as you know that the cells in the G2M phase are the proliferative cells. We check for the expression of the proliferative marker genes, and we find that these marker genes, these cells in the G2M shows higher expression of the expression of the proliferation markers. We performed the differentially expressed genes between the cells, the G1 cells, G2M cells, and the S phase cells. And we find that the cells in the G1, which is this in the middle portion, these cells in the G1 phase, they have distinct gene expression profile compared to the cells in the G1, GS and the G2M. Cells in the GS and the G2M, they showed similar pattern of gene expression profile. The heat map here, the purple color indicates the lower expression, the yellow color indicates the higher expression. So the cells in the G1 shows higher expression of genes that again known to be involved in promoting tumor growth and invasion in multiple cancers, and also involved in the inducing the stemness of the tumors. So thus these cells in the G1 phase also could play a very important role in CCGP tumorigenesis. In conclusion, we have observed a lot of cellular diversity, whether in the proportion of each cell types or in the uh, expression heterogeneity of each cell types within the patient and across the patient. Some particular cell type, some particular cell population, they do express a certain genes, whereas some other population, they express a different set of genes that involve in promoting tumor growth, invasion, and metastasis, etc. And also, there are differences in the profile of the means the gene expression profile of the subpopulation of cells uh, across the patient and even within the patient or within the tumor itself. The malignant cells that we have studied were shown to be enriched with either PMT or fetal cell type signatures, thus suggesting that the partial epithelial to mesenchymal transition and the fetal cellular reprogramming could play a major role in OSCCGP tumorigenesis. And there is a higher infiltration of the immune cells with the T and myeloid cells as the major cell types, indicative of the importance of these T cell and myeloid cells could play a major role in the tumor microenvironment of the OSCCGP tumors. And we also observed there is higher infiltration of the double negative T cells and the intermediate M1 to M2 macrophage polarization OSCCGP. We also saw that there are subpopulation of the malignant cells that even in these treatment naive tumors, there are population of cells that could could involve in tumor growth and invasion and recurrence in OSCCGP. So these studies have recently published in Cancer Science. You may have a look if you are interested. I would like to acknowledge the Heredity Biosciences for conducting such webinar that not only help many students, but also with the different activities that the Biosciences, Heredity Biosciences conducted. I believe that it has made a significant contribution to the science, and also it could significantly made an impact in India. And also, I would like to acknowledge Department of Biotechnology for funding the system medicine cluster studies that we could be able to perform the studies, and also for my DPT Research Associateship and my supervisors in the National Institute of Biomedical Genomics, Professor Arundam Maitra and Professor Partha Majumdar, who guided me throughout these studies, and the clinical collaborators, Dr. Sandeep Ghosh, Paramita Majumdar, provided the samples for our studies, and our team, Shumita, Arunima, Shuproto, Shikhar, and Shumantu, that involved in this study at the National Institute of Biomedical Genomics, and the National Genomics Core, which provide all the facilities required for this study. Thank you.
Thank you, Dr. Sillarine. So it's a wonderful webinar. So it's now open for these discussions. So any of these questions for this audience sites? Any questions from audience sides? Okay, one question from my sides.
Okay.
Yeah. So how does the single cell RNA sequencing differ from the other methods? So understanding from this cellular heterogeneity within a tumor.
Other methods means the bulk methods.
Yes, yes.
Yeah. So the main difference is that with the bulk method, as you know that within the tumors there are lots of different cells that constitute the tumor, the tissue. So each cells they have their own different distinct expression profile. Some cells they have higher expression of a particular gene. Some cells they have lower expression of particular genes. So when we do the bulk, you got the average. So it's not the exact expression level, but with single cells, you will get the expression of all the cells, expression level of all the cells. So you know exactly what are the expression level of that particular genes in that particular tissue.
Okay. So what is the specific advantage of the single cell RNA sequencing? Because this is a
Yeah,
new
here you
company.
can dissect the cellular heterogeneity. You can dissect the cellular heterogeneity not only I mean at a single cell level, not only the gene expression, you can also get the even you could also identify or discover the rare cell types where with bulk sequencing with bulk method, because these rare population they are very small in proportion. When you do the bulk, you will not be able to identify them even if they are present. Like some population in the tumor, like the stem like properties cells, they will be present, the stem cells they will present only a small proportion of those cells, but those cells are really important in again promoting the growth of the tumor later on. Even if after treatment, those particular cells that have stem-like properties, even they present only a small proportion of cells, but then with time they eventually divide and then grow, and then later on they again develop as a big tumor. So these cells we cannot identify by the bulk method, but with the single cell method you could identify them. And you could also not only identify those cells, you can also get the information like the gene expression. You can get even the now we can see the epigenetic information, not only the genomics, if epigenetic, where you can do the attack seek. Even for the T cell example, now the T cell we can able not only the T cell expression, we can get the T cell receptor expression as well as the rearrangement of the VDJ segment of the T cell receptor. And we could identify those T cell properties that they are really effective for this particular. Even we can see that within the tumor, how are the cells interact with each other. This interaction also plays an important role in the tumorigenesis as a whole. So these cells, they have their gene expression profile. They interact with one particular cell type. Some population they have distinct gene expression profile. Some they have distinct gene expression. So these dissecting the heterogeneity itself, it helps a lot, which we cannot be able to obtain with the bulk method.
Okay, okay. So what are the challenges of this tumor heterogeneity?
Challenges of tumor heterogeneity. Challenges of tumor heterogeneity because suppose like a particular tumor, it has a subset of population that do respond to some biological processes going on there, like EMT. and some process is going on which some they do response to some. EMT was also known to be resistance to therapy. To some population of cells, they do respond to therapy. So if the population that are resistant to therapy are present, and the population who responds also are present. So initially when they give the treatment, the patient do responds to treatment, and we think that, oh, this treatment works and this patient will get to recover. But after some time, we'll find that no, this patient again, it relapsed, the cancer relapse again. And then when you give the same treatment, it didn't work anymore, because those cells who respond, it already died. But those cells who are resistant to that particular treatments, they keep growing. So it's not responding anymore. They are resistant to that particular treatment. And then the way they grow also, like they do evolve eventually with time, and to escape even from the immune response itself inside the body, they became immunosuppressive. There is the environment there became immunosuppressive, and the tumor cells, they find their way to escape the treatment with time. So this one we cannot really, this is still very challenging now, because we captured the single cell, even we captured as like a one time captured, but then eventually after some time, those cells again, they do change, evolve, and then, so it's still very challenging. But comparatively to the previous method, single cell, it has helped better than the previous method.
Okay, thank you. For this last question from audience sides. So, for a student and researcher, if they are interested for this field, what you have to advise for the pitching for this study for tumor heterogeneity using single cell RNA sequencing techniques?
I didn't. What will
If
I
any,
suggest? Yeah.
if anyone to interested for this field, so what they will be doing for that in the studying purpose, the tumor
Or
heterogeneity using this? What you
how
have to
to
suggest?
prepare?
How to suggest to the students or researchers? So they will be implementing for this research to his or her career.
Okay. Yeah. Yeah. This is the emerging field that people now are really jump into this technology, even though it's still very costly, but I think it will make a difference, because you'll be able to discover more new things, and which with the previous method you cannot discover. So I would suggest that if you have something which you really cannot do with the bulk method, you want to implement this single cell method, in the sense that for a particular complex tissue, you really want to you really want to do studies, although it's a very costly. This is, the cost is still very high, but if you can afford that cost, or maybe apply for some fellowship or grant, if you can write a good proposal, if you really have that good idea, and then projected that with without this technology you cannot achieve that goal, then maybe you can get the funding for your study.
Yes,
Because
yes,
the main challenges is the funding, because this is very costly, and if you do not get the fund, we cannot do. But if that is not possible, then you can train yourself in the analysis part, where you can download the available data set. There are lots of available data set freely available, or you can just contact the corresponding author. But there are many available data set. You can use those data set and then explore those data set. You can analyze them and then validate in validate in the lab. That will be cost effective. But if you have your own sample, that will be better. But since the cost is high, you need to have a good planning, a good goal, where you really need to achieve, when you can achieve only by doing single cell sequencing. If your idea is strong enough, and the planning and the goal is really strong, definitely you'll get the funding and you can do by
The
yourself.
sample receiving is a very challenging part also.
Sample processing is very challenging. I have done it from the scratch. I know how much challenging it is. One sample is very different from the other sample, and one sample from one tissue type is very different from the sample from other tissue type. It's very challenging, and because we need to and then it's not like every institute they have the hospital very near. You cannot get the sample because the time is very important here, because we have to maintain live cells, live
Mhm.
healthy cells. Otherwise then the gene expression whichever you got, it's not the actual gene expression because it has changed throughout the time, and during the process, while processing the cells undergoing lots of stress, so they change the gene expression also. So
Yes,
to really get the exact accurate gene expression, we have to maintain the time, maintain the temperature, and get healthy cells. So to get the healthy cell is very challenging. We have
yes,
to train ourselves that, and then train with different tissue, because one tissue is behave very differently from the other tissues. The easiest one is the PBMCs, which is that is much more easier, but from the tissue is much challenging because there are lots of fibers, there are
mhm.
many things, you can get lots of debris and all during preparation. So it's very tricky, but it's not impossible.
Yes, yes.
Yeah.
So thank you very much, Dr. Silan. So first, one or two questions from the audience sides. Anyone from this asking for the any questions for the audience sides? Only one or two questions. Anyone, or else we will be end of the session. Okay. Okay. Probably every and they all are the students, no? So they will be little understand. But I really appreciated huge work you did in your career, and I'm really appreciated. So finally, audience, we will be end of this session. So, on behalf of Heredity Biosciences team, I extend my heartfelt gratitude to each and everyone of you for being a part of this impactful webinar. Today's webinar is a remarkable journey into this topic, characterization of tumor cellular heterogeneity using single-cell RNA sequencing. Thank you, Dr. Sillarine, for your expertise and knowledge in the field of cancer biology, and I really appreciated for this for your work. It's a very delicious work also, because we had a lot of efforts for this field, and truly commendable. And we are really honored for have a opportunity to speak about this thoughts in your experimental thoughts to the audience. I am personally learning a lot for this webinar, and your presentation has been enlightening, offering valuable insight to the significant to the patients and to the research and diagnosis purpose, also advancing to the cancer research part also. Thank you very much again,
Thank
and
you very much for the opportunity. I'm very glad.
definitely for the future we will be any collaborations, any
Yeah,
things we will do for that. Okay.
sure. Sure.
So, I would like to acknowledge to all these members to present here to this webinar, all these participants who will actively engage to this webinar. Hopefully, all these participants to be understand to that. I am also learning a lot. Probably all will be learning a lot for this webinar. Your response and thoughtful questions. I am asking for the questions the audience sides. So this today's discussions made the event is success, and the final questions the audience sides will convey to everyone to that. This any one of the questions to that I will be share to her mail ID, Dr. Salvi's mail ID is to everyone.
Sure.
Everyone should do that. So any of these questions for this future, so you will be mailed to her also. Okay.
Sure. Sure.
Mainly, I acknowledge my team members, dedicated team members, Susri, Sriram, and other members also, Heredity Biosciences. They are totally engaged to my speaker, today's speaker. They always connected last 1 month, last nine already decided to the date, and we are extended to the 16th because of the some of the official issues happening. But really, Dr. Sillarine, you are truly waiting for this day, and finally we are successfully conducted. So this is really amazing, and your side. Thank you again for that, for my side and my team side, to given the delivery of the talk also in this excellent this topic, and you have the webinar. And once again, thank you very much, Dr. Celine, for the sharing your expertise with us, and thank you all participants making this webinar. To round success, I look forward to the hosting more event in this future and to the experts. Thank you very much for this wonderful day. Thank you very much.